So, inbreeding brought us wonderful results in creating and improving breeds. So, what we did it for, what we aimed at, that was fix desired features in animals and make sure they breed through for that. Well, we succeeded in that. With our combination of inbreeding and selection, we improved breeds. But at the same time, what we have seen is that any overdoing of inbreeding makes it a threat for the future of our breeds. It harms the vitality of the animals. Look at lifespan. There are breeds who live only half a dog's life, well, the average age they reach is about 6, 7 or 8 years. So that's one aspect. Another aspect is what we talked about, the consequences for fertility. So if we apply inbreeding as a tool, then we should be very careful. We shouldn't take any risk. We should be aware that selection against genetic disorders, it's a common belief among dog breeders. Well, we apply inbreeding, and on the other hand we take away the negative effects of it by selection. By selection you can only act against the disorders you have seen. You can only act against the bad effects of the inbreeding you see in your litters, in your animals. What you can't compensate for is all the defects you don't see at this moment in your breeding stock. You can't compensate for the loss of vitality. Loss of vitality is a very slow process that goes on generation after generation, and there is no way you can measure it, see it as an individual breeder. You can see it and measure it if you look at what happens to a breed stock, a breed population as a whole. So there is absolutely no way to select and compensate for the negative effects in all 25,000 pairs of genes. Well, the main cause of an increasing coefficient of inbreeding is the overuse of male breeding stock. So there are too few males, too few dogs used in most breed populations. On the side of the females, bitches, well, most of the time you need enough animals to keep up for your genetic, to have enough genetic variation in it. But on the male side, we select our absolutely top breeding animals and then get too many litters, too many offspring, which means that 3 to 4 generations later, the offspring of those overused males will meet and show the genetic problems they brought into your population. Well, it's not, as you can see, it's not a new, really new problem. Also Noah faced it and talked to that little male that was overused a little bit. Our best solution in terms of applying inbreeding as breeding tool is called prevention. The more we limit the increase of the level of inbreeding, the better our chances are that natural selection compensates for it. You should realize that in any population, any closed population, any species, I would say, there is always a certain amount of inbreeding. You can't avoid inbreeding at all. Inbreeding is part of life as we know it. But by applying in our breed populations the slow increase each generation, natural selection can compensate for what goes wrong. There's a general rule: you shouldn't have an increase in your coefficient of inbreeding all over the population of more than 1%. Well, if you succeed in limiting that increase to that level, but then you should do that as the group of breeders as a whole, then your population, your breed population, your breed will survive. If we don't put restrictions to inbreeding in our breeding programs, in the end, inbreeding will restrict us in our breeding programs. So it's one way or another, or we are very careful in what we do, or if not, then sooner or later we get the problems I showed you in the slides with the examples. I realize that the things I explained about inbreeding are not the things that are a common belief among dog breeders, also cat breeders. Well, people have been brought up in dog breeding with an absolutely belief and absolutely trust and absolute confidence in the benefits of inbreeding. There was not too much attention for the negative effect of inbreeding, and so I think I should be clear about this. It's of course up to you to decide in how you act within your breeding and selection programs. I want to stress I can only ring the bell in telling you what happens, what can happen, and what it might lead to. I thank you for your attention. I'm going to tell you something about the developments in DNA. People, dog breeders, expect a lot from what DNA could bring them as benefits, and it's quite important that you realize what's available and what's not. If we look at what happened between 1900 and 2000 on the level of DNA, then in 1900 we started with our first microscopes, and we saw when cells were dividing during cell division, we saw that the nucleus of the cells fell apart, and there were strings you could call, and we called them chromosomes. Now, 100 years later, we in the meantime found out what these chromosomes are, and we get complicated structures, that complicated molecules where all the genetic information is stored. I would like to give you a 10-minute course on DNA so that you have an idea what's going on, what these people who promote DNA tests and promote all kinds of tests, what they are talking about. It's in fact quite simple. Each chromosome is a long molecule of DNA. It's a chain that with 4 different elements in it: adenine, cytosine, thymine, and guanine. In fact, the language of life only uses 4 characters. We call them A, C, T, and G. If you compare it with the language we speak, we use 26 characters and maybe even more. The language of life only speaks in 3 letter words, codons we call them. Look at our language, how complicated it is, with words of different length and all kinds of combinations of characters. So the structure of DNA, the structure we see here, is in fact just a long chain of only 4 different characters and only that you can read in 3-letter words. Well, then you need something to know what you're reading. So each codon codes for an amino acid. And in the translation process, the chromosome, the DNA, is translated into a chain of amino acids. That chain of amino acids is what we call proteins. And that protein, again, is part of an enzyme. So a gene is a chain of codons coding for the construction, for the building of a protein. A gene is nothing more and nothing less than a building plan for a protein. Well, I told you we do have 25,000 pairs of genes, so the building plan of life, is a building plan consisting of the possibility to build 25,000 different enzymes. If I look at an example, if you are reading the DNA in a certain area on a chromosome, then you could see a code like this. I told you, only 4 different letters. If you are going to read this, you should read it in groups of 3, so in codons. And if you have your translation handbook for codons, then you know that this piece of DNA is coding for leucine, histidine, alanine, lysine, valine. So it's coding for a chain of amino acids. In this way, hundreds of codons are translated into chains of amino acids and are forming proteins. If you then look at the building plan, the codes of a gene, then you can see, first of all, it's not more complicated, it's just more. Instead of the 10 or 12 characters we had in the example we used, now we have here the blueprint for a protein consisting of 257 amino acids. This is just the building plan for a protein. Well, that looks complicated, it looks like it's difficult, but in fact it isn't. It is a quite simple system, a quite simple language of life in translating codes into functions. What are mutations? Mutations are just errors in the genetic code. One or more codons have been changed or maybe are missing. Change in a codon or a missing codon causes a change in an enzyme resulting from it. If you miss codons or you have different codons in your blueprint, then it means that you get different amino acids built in your protein, or maybe parts of the protein are missing, which means that you get consequences for the enzyme that's formed out of that protein chain. Which means that in the end, that if an enzyme is changed, it means that a function maybe is not fulfilled within the body. It means that a function is fulfilled in a different way. And what you can see is that there are different levels of damage to enzymes. In some cases, the enzyme is not formed at all. In other cases, the enzyme exists but has a somewhat different function or not the quality of the function it should have. And in some cases also mutations exist where in fact there is no phenotypic